Hi, today I'll be talking about a paper by Dr. Rafael Gaveo and corresponding author Dr. Min Chen. This paper is about photoactive phytochromes, and they interact with a protein that they call HEMERA to establish photomorphogenesis in Arabidopsis. So the picture they paint in the beginning is a commonly understood mechanism of phytochrome. Phytochrome senses the light and then turns active, and then it causes a signaling cascade which leads to photomorphogenesis in the plant, which is basically the plant stops growing and it starts growing leaves in order to begin its life inside the light. So the mystery they want to understand is some of the early stages of this signaling cascade, and they propose that there's this molecule called HEMERA that acts very early in this process. Here they have a immunoblot of HEMERA with the Arabidopsis at different light levels. So in 4 days of true dark or red or far red light, we see HEMERA, there's more HEMERA protein in the light conditions than the dark conditions. Meanwhile, in those same conditions, the RNA level is about the same. So this suggests that the amount of HEMERA in the cell is regulated more post-transcription and physiological conditions rather than genetic conditions. This figure is kind of showing the same thing, where the RNA level is very high, but the protein level is about very similar. And also, they kind of talk about this later in the paper, but this suggests that the HEMERA protein is being rapidly degraded, and it kind of stabilizes in the presence of light. And the process of this degradation is kind of left a mystery in this particular paper. So in this figure, they want to investigate if phytochrome is involved in the process. And they have some phytochrome mutants here, phytochrome A and phytochrome B, so that they don't respond to far red light or red light like they used to. And you see in the case where you show phytochrome, you show far red light to phytochrome A, HEMERA does not accumulate. So this kind of shows a interaction between phytochrome and HEMERA, where phytochrome is not working, then HEMERA itself also does not accumulate. And that kind of shows that HEMERA is not directly responding to light, it's directly responding to the active phytochrome. So now that we know that HEMERA accumulates in response to light and it accumulates in response to the active phytochrome, we want to see if they interact more directly themselves. And this is what this figure is kind of showing. They use what they call a GST pull down assay, and they show that the HEMERA does bind to the active phytochrome, and it binds more to the active phytochrome than the inactive phytochrome, which is what this blot shows right here. And so it's basically showing that they do indeed bind, at least in the test tube. And then they go on to test to see if they bind in vivo inside the cell using a immunoblot. And basically, just based on this one band here, it shows that they're showing that they're able to pool the same molecule using an HMR tag and a phytochrome tag. So it's kind of showing the HMR and the phytochrome are indeed bound together inside the cell. So now that they've established some kind of direct interaction between HEMERA and phytochrome, they want to dig a little more biochemical to see what parts of the phytochrome does HEMERA bind to. And here phytochrome A, they section off phytochrome into different parts, the N section, the C section, and then the small section in between that's directly responsible for sensing light. And then they use another GST assay, and they see that HMR does indeed bind to the N section, and particularly it binds to the GP section, the section that senses light. So again, it's kind of showing, this is kind of just a little bit more biochemical picture of how HEMERA is binding to the phytochrome. Then they go on to dissect HEMERA, and they pull it into all these different sections here. And they do another GST assay, and they see that the phytochrome binds to the PIR1 section, as well as the PIR2 section, but it binds a little bit stronger to the PIR1 section. So now that after digging a little bit more biochemically, here we see that they go on to investigate more mutants of HEMERA to see if the mutants of HEMERA really affect the phenotype. And here we have various mutants of HEMERA, and they all show very low accumulations of HEMERA in the presence of light. So even a small mutation in the HEMERA kind of messes up the whole process, basically, is what this figure is saying here. Again, they also add another mutation where even if the HEMERA is really mutated, the phytochrome itself is very strong. So they want to see if the phytochrome kind of wins out over the mutated HEMERA. And based on this band here, they see that HEMERA does accumulate to a small amount when we have a particularly strong or active phytochrome binding to it. And basically, you can also see the phenotype here, where in the HMR mutant, it doesn't really respond to light, and in the phytochrome mutant, it responds to light prematurely. And when you combine them, you kind of get these little messed up figures, but they do respond to light at least at some point in between. Basically, these two figures kind of show that the active binding between HEMERA and phytochrome is more important to leading to the whole phenotype than the exact chemical makeup of HEMERA by itself. So it's just a lot of emphasis on the binding. And then in this last figure here, they finally wanted to include the PIF, the phytochrome interacting factors. They note in the paper that these factors kind of are repressing certain genes that handle photomorphogenesis. And they know that when the plant senses light, these particular factors get degraded. So they want to see if there's some correlation of degradation between HEMERA and phytochrome. And indeed, we see in this plot here that whenever HEMERA is absent, the PIF is present, and when HEMERA accumulates, the PIF is gone. So it seems to show that when HEMERA doesn't accumulate in response to light, the PIFs are also degraded. In this figure here, they basically did the same thing, but with every single PIF gene they could think of. So we get like about 12 of these here. And the trend is basically the same, where in conditions where HEMERA does accumulate, the genes themselves are either expressed or repressed based on the presence of PIF. So when HEMERA accumulates, these interacting factors are degraded, and the genes that they either repress or induce are expressed or unexpressed respectively. And the whole picture is basically how the accumulation of HEMERA breaks down the interacting factors. So this is the overall picture they give at the end of the paper. And really what they introduce to the common understanding is the importance of this molecule HEMERA right here, where they show that really early in the whole signaling cascade, HEMERA is involved. It binds directly to the activated vitochrome, and that's kind of like the second step, after which everything else follows. It's such an important step that even small mix-ups in this protein kind of stop the entire process. So that's what this paper really introduced to the understanding of this kind of phenomenon here. They go on to talk about more downstream actions with the interacting factors. How do they exactly get degraded? Are they degraded by the photobody or by something else? They say this is still a mystery that they'll have to investigate later on. But what is really solidly established is basically the work of this protein here at HEMERA. And that's what Dr. Min Chen and Dr. Galfeo's paper really proved in this experiment.